Hello, and welcome to all of you for joining us uh, in this Back Ashore series, a new series from Schmidt Ocean Institute, where we invite scientists who have sailed on Falkor to discuss the outcomes of their science. Thank you, everyone, for joining us. My name is Allison Miller. I'm the Senior Manager of Research Programs at Schmidt Ocean Institute, and today I am joined by four scientists that sailed on board Falkor in 2019 to the coast of Costa Rica to explore the deep sea waters, off the Costa Rican margin. Today, I'm joined by Eric Cortez from Temple University, who was the chief scientist on board this expedition on Falcor, Doctors Lisa Levin and Greg Rouse from Scripps Institution of Oceanography at the University of California, San Diego, and recently retired Dr. Jorge Cortez from the University of Costa Rica. Thank you all for joining us. I hope that footage that we just watched reminded everyone of the wondrous animals and organisms and life on the deep sea floors of Costa Rica that we witnessed in 2019. And I'm really looking forward to hearing about what you guys have been up to since and some of the analysis and research taking place. So just to refresh our audience, the goals of the expedition was to characterize seamounts and seeps around the Costa Rican margin and to examine the chemical and biological connections of the region in order to establish baseline knowledge. And in very exciting news, just late last year, President Carlos Alvarado of Costa Rica signed a decree expanding the Cocos Islands National Park where this expedition took place, and we'll be talking more about that during our discussion. So Eric, I'd like to start with you as the chief scientist of this expedition. Could you tell us what the motivation was for putting together this proposal to SOI and what you found once you got on site?
Sure. Thanks for having us here. This is it's fun to relive the expedition. Just watching those videos brought back a lot of memories. So it was part of a larger project that really Lisa and Greg started over 10 years ago. But the funding that we had from the National Science Foundation was to look at the seeps, the cold seeps near the subduction zone off the west coast of Costa Rica, and look at their change over time and how succession kind of works there. And we did some of that work. We continued some of that work on the Falcor, but the real motivation for coming back with the Falcor was really to do more exploration, something that the National Science Foundation doesn't necessarily fund all the time. And we were really interested in looking further offshore at some of the seamounts on the way out to Cocos Island, as the conservation work, the planning to get the expansion that just occurred was really underway. And after talking to Jorge and some more of the scientists down there at University of Costa Rica and the students, that seemed like the biggest information need for them at the time. And so we wrote to the Schmidt Ocean Institute and were awarded this cruise on the Falcor. And I think, I know Jorge will talk a lot more about this, but I think it really went a long way towards helping the efforts to make these seamounts a protected area.
Excellent. Yes, exploration is always the first step in a lot of these endeavors, so I'm glad we could help out. Could you tell us about some of the analysis that has taken place since you returned back to shore?
Yeah, I mean, science isn't an overnight endeavor. So we've been working a lot on the distribution of deep sea corals on these seamounts, and then further out into the Pacific, we found some really interesting connections between these seamounts and those near the equator, extending pretty far across the Pacific, which is something we were kind of surprised about. I didn't really imagine, and a lot of them, the sort of biogeographic maps don't connect those areas very well. So I was a little surprised about that, and really having the other time point, another year of field work to go out there and look at some of the seeps that are in some of they look like those scars in this map that you're looking at right now. Seeing some of those seeps and being able to extend our time series has really helped us get a better picture of the succession and the time scale for change in the deep ocean that we really rarely get a glimpse at.
Great. Well, I'm glad to hear that you can still go to sea and find things that surprise you. So that's really the spirit of exploration. Jorge, we've just mentioned some of the Cocos Islands expansion that was decreed last year. But this was not your first time going there, and I know you've spent a lot of your career studying that area. Could you tell us more about what makes this area so special?
Well, Isla del Coco, as we call it in Spanish, is 500 kilometers offshore. So it takes a day and a half to get there. And because of the island, Costa Rica has a huge marine area. 92% of this country is marine. And we're just starting to know what's in there. And this expedition was great. It was with the previous expeditions, we saw the seeps and the continental margin, which was very, very interesting. And a couple of seamounts close to shore. But with the Falkor expedition, we went all the way out to the island and past the island, saw all these other seamounts. And in that video that we saw at the beginning, it was just amazing. And it had a big impact here in Costa Rica. I've shown it to many different groups of people, including government officials, and no one imagined that we had all that within our country. So that's the value of this type of expeditions, especially for small countries like Costa Rica, which doesn't have the capability of going out there.
Thanks, Jorge. And when you and I were planning for this session over email, you also talked about how the opportunity to bring some of the dignitaries and decision makers on board the ship before we left port may have also played an important role in allowing others that are not scientists but yet are making these decisions to be motivated and inspired also by your work. Could you talk a little bit about those opportunities, and if there's anything else you wanted to add about how some of the data you collected might have motivated and inspired the expansion as well?
Yes. This project of expansion of the protected area around Isla Coco started in 2009 with a Pristine Seas National Geographic expedition. So it's been a long process. It took 12 years to get to signing this expansion that we're seeing on our screen right now. The black circle was the previous totally protected area, and then the black polygon was an expansion that included some seamounts. And with the signature last year by President Alvarado, the blue square, the blue rectangle is now the Isla del Coco National Park. So it's no take in that area. And the red polygon is a special management area. We had the opportunity of having the Vice Minister of the Environment, Aide Rodriguez, on board, which was very active in promoting this and the creation of this expansion. And she was very impressed of the vessel, of the crew, and all the scientists and all the work that was being done.
Excellent. Thank you so much. Lisa, I think Jorge set you up great to talk about some of your work. You are the co-founder of both the Deep Ocean Observing Strategy and the Deep Ocean Stewardship Initiative. And so a lot of your work now is focused on bringing science, and specifically science in the deep sea, to these decision makers to help protect and procure the fate of the deep sea ecosystems. Could you tell us a little bit more about your work and how the expansion of Cocos Island National Park may have set a good example in the international arena for your work?
Sure, thank you. Well, from my perspective as an ecologist, this expansion protects a really critical hotspot of biodiversity, and it provides safe habitat to a whole series of highly migratory species and top predators that move through Costa Rica. And these animals then distribute the ecosystem benefits across the entire Eastern Tropical Pacific region, and sometimes even far beyond that. And my colleague, Melania Guerra, who's a Costa Rican policy expert, tells me that the domestic policy process that created the MPA that we're talking about followed scientific evidence that determined the importance of these seamounts in the life cycle of these highly migratory species, and that I believe this provides a really valuable model to other countries of how science can inform policy. And then in terms of diplomacy, it's notable that this MPA is a Costa Rican component of what's a much larger Eastern Tropical Pacific initiative called CMAR that involves Colombia and Panama and Ecuador and Costa Rica. And my understanding is that this is maybe the first time that that these countries, or maybe any countries, I'm not quite sure, with connecting maritime borders have joined together to create a single public policy. So this sets a really important example for regional transboundary MPAs. And then finally, I think it's worth noting that the creation of this MPA allows Costa Rica to reach its conservation goal of conserving 30% of its ocean territory. In other words, this blue target of 30 by 30. And again, Costa Rica is a great model that could be replicated around the world.
Certainly, and congratulations to the country of Costa Rica for reaching that goal. I'm loving all of the imagery that we're seeing as Lisa speaks about the importance of protection. Lisa, you've also sailed on Falkor many times, kind of wearing a hat of looking at biodiversity through the lens of conservation and how important it is to understand these regions and the biodiversity that lives in them, again, so that we can protect them and better understand what needs to be conserved and the value of these deep sea ecosystems. And I was wondering if you wanted to talk a little bit about that experience, especially in the context of this expedition.
Well, biodiversity underpins pretty much all of the ecosystem services that are provided by the ocean that we rely on to keep this planet healthy. And, just to give some examples, the diversity of phytoplankton remove carbon dioxide from the atmosphere, and the diversity of animals that are in the ocean transport that carbon vertically and down into the deep sea. And then on the bottom, they help sequester it. And at methane seeps, for example, like the ones we've been studying off Costa Rica, there are a whole series of animals and microbes that lock up methane and keep it from reaching the atmosphere. So that's a really important service. And then we know that biodiversity fuels the food webs that generate our global fisheries, and its biodiversity is the source of new industrial agents and new bio-inspiration for things like bone grafts and fiber optics and armor and much more. And then, of course, for biopharmaceuticals that are, helping to cure human ailments like cancer and COVID and so on. So it's really important.
Definitely. Thank you. And last but not least, Greg, you sailed as an invertebrate taxonomist aboard this expedition, and you have sailed in that role on board many expeditions on board Falkor. And I know you have named and described a few new species from this expedition, and you have a long list of ones still to check out. But before you reveal the exciting new species you've discovered, I was wondering if you could share with our viewers a little bit about what is actually involved in describing and learning about a new species once you're back in the lab.
Yeah, thanks, Allison. I think one point to be made to the audience is how much of the world's biodiversity, and particularly ocean biodiversity, is relatively undiscovered. And we might have seen them or noted them, but many things, many species, we'll call them, don't actually have names. So that is one of my roles, is to actually provide names that allows them to really be recognized and to become part of science by ecologists such as Lisa and Eric and Jorge, and to really be counted when we do biodiversity surveys. And if things don't have a name, it's really can be problematic. So I really think it's important to give these animals and plants names. The trick is that it's often quite a long process. There's a series of rules involved in naming species. And one of the big challenges is often the burden of history. So many collections over the years have named species. And if you look in the records, there are about 400,000 marine animal species that have been named. But when people go through and review these names, often many species have been named twice or 3 times or even 4 times, particularly in shallow marine waters. And this leads to this problem of when you find things, not so much a problem in the deep sea, but even then it can be a problem, is have you found something that's new, or is it something that was discovered 100 years ago? And that's the burden on us, is to really prove that we have found something new. These days, we really have a tool that is very helpful, apart from the wonderful imagery that the submersible Sebastian provides us, and being able to collect these animals in beautiful condition to bring them up for study and photography. We can then sequence their DNA. And that is a fantastic tool for really delimiting and helping us figure out if we have a new species. The trouble is with the old discoveries is that are sitting in museum collections, we often can't sequence their DNA and prove that we have something new. So sometimes we're kind of stuck and we can't decide if we have a new species, but in the deep sea, we often are able to really prove that we have something new. And then the task is to take a specimen, sequence its DNA. We would then lodge it in a museum. We have a museum collection here at Scripps, and we also lodge and have another set of collection that's at the Zoological Museum at the University of Costa Rica in relation to this particular project. And those are important, these archives. These specimens are then held there for future science to refer to, and we think that's a really important part of the process. Then we have to go through the process of naming the species, which means it's a peer-reviewed scientific publication, and you follow a set of rules, but for animals is called the International Code of Zoological Nomenclature. And that ends up with a binomial name. Our binomial name is Homo sapiens. And so we end up giving binomial names to these new species and then putting them into context of the other general groups that they might belong to. Does that give the basic picture that you were thinking of, Allison?
Yes, yes, very arduous and detailed process, but thank you for sharing.
We were talking before about the timeline from discovery of or sampling of a specimen to the naming of a species, and Jorge had read that the average time can be about 21 years, and I wasn't too surprised at that number. I know even in my lab, we often have timelines that are well over 5 to 10 years. And with reference to Costa Rica, me, Lisa, and Jorge and Eric have been visiting the inner margin, the continental margin of Costa Rica, where these former seamounts are going in under, they're subducting under the mainland of Costa Rica, and that's generating these methane seeps. And here you can see the top of Mound 12 that's relatively near shore of Costa Rica, and it's an active methane seep. And there you can see carbonate rock, as well as lots of animals. And there are several of the new species we've described. A lot of the new species that have come out of our Costa Rica work are from this inner margin. And we're up to 37 new species so far of animals, with quite a few still in the works. And in this picture, you can see several new species of mussels. There are 2 new species at the top of Mound 12. And there's the iconic, if we can go to the next one, Hannah. Here you can see a close-up showing those 2 species. One is kind of yellowy and the other one's more brown. And they're in a genus called Bathymodiolus, but you can also see there a shrimp. That was a new species of shrimp that's sitting there. And those little limpets and little snails that are sitting on there, I know Eric's lab are working on those, and there are potential new species sitting in there. Keep going, please. Here we see a microbial mat that's actually at the deeper part of the seeps off Costa Rica at a place we call Scar. And there you can see white microbial mat covered with little annelid worms. And this group, Lisa and I have worked on a lot. They graze on microbial mats and are found very commonly at methane seeps and hydrothermal vents around the world. And this is a new species that we're about to name for a German colleague of ours. And it's in the genus called Ophriotrocha. The next one is, this is an amazing story. We had seen this animal way back in 2009 and 10. It was kind of a mystery animal. It was gliding just above the surface of the mud. We would see it routinely, but it was very difficult to catch. It needed to be caught with a suction sampler, and the animals were pretty mobile. And finally, this is an image from the 2019 cruise, but we did catch them on another cruise. And this is a male that's swimming, looking for a female to mate with. It's another kind of polychaete, and we're really looking forward to naming that one this year. It's one of our new ones to be named, and quite an interesting story about the natural history of this animal that we've got coming together. The females live in burrows and the males fly around like flying carpets looking for the females.
Wow.
That's going to be a new genus and named for a famous pilot of the human-occupied vehicle Alvin, Bruce Strickrott, who was the first person to ever catch one successfully.
Awesome,
Next,
thanks.
And here we're at the bottom of Scar, 1,800 meters down. And this is another couple of new species here if we zoom into the next slide. These are a feather duster worm that you can see there with its feathery crown. Next to the white shells are giant clams that have symbiotic bacteria in them. And next to them are some tube worms that also use symbiotic bacteria. Both the clam and the tube worm are actually well-known species from seeps along the margin of the Pacific. Around the tube worm, though, you can see those little orange kind of threads. Those are a new species of ribbon worm. That's because of its association with that tube worm Escarpia. We're naming that after the tube worm. And then there are those beautiful feathers you can see there. That's a calcareous tube worm called a sepulid. And this led to a really amazing study by our other collaborator who isn't on this call, Shana Gafridi from Occidental College. These are a tube worm that also uses symbiotic bacteria. Methane oxidizing bacteria are found all over its feathery crown. And that was published just last year. And then we also named this as a new species last year as well in the genus Laminatubus. And then really the cruise that we're talking about today, we went further out after the Costa Rica margin to the seamounts offshore that have yet to subduct under Costa Rica. And I have to say this is really a work in progress for us in that there are so many new species that we need to find experts to help us. A lot of them are bigger animals that we collected. And here you can see the manipulator arm grabbing a carnivorous sponge that has those 4 bands of sticky spikes coming out along its margin. But what we were interested in, as well as the carnivorous sponge, which we did collect, are the 2 beautiful blue-purple worms that you can see there on it. And this was really intrigued the Sebastian pilots, in particular J-Rod. And this is actually a male and a female pair that we did collect. And we called this the ice dragon worm. This cruise was at the end period of the Game of Thrones TV series that have everyone enthralled. And so we decided the nickname for this worm was the ice dragon worm. And one of my PhD students, Avery Hiley, is in the process of writing this up as it's going to be a really fun new species description. Here you can see them brought up to the surface sitting in the lab. We give them a number, they get a name for processing through to the DNA phase and all of the other work that we do on it. Eventually, one of those specimens will be retained here at Scripps and the other one will be sent to the Zoological Museum at the University of Costa Rica. We haven't decided on the name of this one, but the DNA sequencing is telling us that this is a new genus as well as a new species. And we have many others still in the works that will be coming out, no doubt, over the next decade, I would say.
Thanks, Greg. Yeah, we were speaking before we went live about the length of time it takes to just analyze everything that the four of you are working and I just wanted to remind everyone, I know you can do the math, but we're just 3 years after this expedition took place at sea, and there's still a lot going on. So I'm looking forward to even hearing more from you in the future, but this was a great look back at what you all are doing. I know you've been really busy. I wanted to remind the audience to feel free to put in some questions for our scientists in the chat. They would love to tell you more about their work. Before we go to the audience questions, I would be remiss if we didn't go back to our time on Falkor and ask you all, what was your favorite moment while at sea on board the expedition? And I'm going to start with you, Lisa.
Okay. Well, I had 1,000 favorite moments. It was just an amazing expedition, and it really, we really can't pick one. But if you could show the slides I sent you, I thought I would highlight a few things that we found that reflect what I'll call benthic pelagic coupling, because I always talk about how connected the deep sea is to shallow water, and we were really able to see that at our shallowest site, Cocos. It also sits right in the middle of oxygen minimum zones, and those of you who know me know I'm really interested in low oxygen environments. So I would say this movie that maybe you can just show it again, put us straight in the middle of a cutlass fish school at 400 meters. I took this with my cell phone, actually, but it was such an amazing feeling to be surrounded by this school of fish and to be able to be right there with the Falkor camera. So that's one of my real highlights from this cruise. But there are a couple other things. Let's see what the next, finding. And I really, it was thanks to Eric's Sentry work. We discovered that these tuna crabs migrate from the bottom to the top. And so they cover the bottom, they'll feed on the bottom, they'll live down there, and then they'll bring that energy back up to the surface. So that's another great example of, and they seem to have day-night migration. So that's another great example of benthic pelagic coupling. And then the last thing wasn't from Cocos, it was actually the finding that Xenophiophores, which are giant protozoans, this sediment agglutinated single-celled organism that you see up in the left corner there, that's about the size of a fish, of a fist. We discovered that snailfish lay their eggs in there. We found the embryos, and Greg was able to sequence them and identify at least the fish genus. And in fact, we found two different Xenophiophores with embryos of two different types of snailfish, and finding that single-cell animals can be a nursery for vertebrates. It was just amazing to me. And I stuck another great nursery picture in, and there's a black coral there in the lower left that's a nursery habitat for very, very, very tiny galatheid crabs, some just a millimeter in size. And the amazing Falkor cameras were able to zoom in on and show us these, these nursery functions in a way that, I've never been able to see before.
Thanks, Lisa. I love your excitement is coming through even through Zoom 3 years later. So thank you for sharing that. Jorge, what was your favorite moment on board Falkor?
Just as Lisa said, the entire expedition was a great experience. The vessel, the crew, Sebastian, our ROV, they're great. The cameras that the ROV has are amazing. So, and it was just beautiful to see the panoramic view of some of these creatures, and then zoom in and see very, very in great detail all these creatures. And you've been seeing all these pictures and all the new species that are coming. It was great. It was excellent expeditions. All of my colleagues here, it was great to be at sea with them, and I learned a lot. I've been working in shallow water, so I learned a lot and really opened my eyes to the deep regions of this country. And that's the talk I've been giving to everyone that wants to listen to it here about that. And I titled this presentation is the deep regions of Costa Rica, an unknown country. And it's been very impacting. And I think that was, for me, that was the highlight of being on this expedition. Here we see the Falkor at Isla del Coco. We visited the island and went on shore and talked with the park rangers and did some walking on the island.
Yeah, I love this image that you're seeing now, and thank you for sharing that, Jorge. Eric, what was your favorite moment of the cruise or favorite moments?
I mean, the cutlass fish video is really amazing. And I just remember everyone flooding into the control van and just oohing and eyeing. And it was really an amazing moment for all of us to be in there and be there with the pilots and the crew and the technicians. And, you know, everybody kind of came in and shared in that moment. But, you know, a few of the things that I really remember are some of the interactions with the people of Costa Rica that we had, the visit to the island. Yeah, this moment was really just phenomenal. But the visit to the island and talking with some of the rangers and being able to show them the deeper parts of the park that they're taking care of and share some of the research with them, and meeting with the vice minister of the environment and being able to host them on board was really fantastic. Being able to share some of this information and knowing that our exploration was going to help them and feed into those efforts. And Jorge organized a symposium with a bunch of students from the University of Costa Rica, and we got to share some of our research with them, and we got to hear a lot about their research. And, you know, we got to bring some students from Costa Rica out to sea with us, and they got to have their first experience with ROVs and really deep water work. And, you know, it's those, I think those cultural connections that we were able to make was really, really rewarding, really just knowing that our research was gonna go somewhere and was gonna help their efforts was really fantastic. And that's, you know, that's something that I'll definitely always remember about the trip.
Thanks for sharing, Eric. Yeah, we always have to remember the personal side of science and bringing those stories back to the people whose coasts we're doing work on. That's a really important aspect. Greg, would you like to share what your favorite moment was?
This was a really unique cruise in many ways, in that we were targeting a whole lot of sites one after another. So the idea was to go out from the coast to end up at Cocos and visit these seamounts along the way, one day at a time, pretty much. And so that was really incredible that every day you're on a new place, and these seamounts come up to different heights, in a sense, below the surface. So they were all summiting at different depths. And if you can imagine thinking of a mountain on the land, that it has zonation of vegetation as you go up it. Well, we were doing transects up the side of these seamounts over hundreds of meters. And as you go we would start low down and come up to usually sometimes near the top of the seamount and traverse through lots of different water parameters, such as particularly oxygen. And so the seamount slopes were really dramatically different. And they all differed from each other. There were some shared similarities, but every day was so wonderfully different. And I think often, me and Lisa, I know, was particularly excited every day when we would journey through the oxygen minimum zone and just see this remarkable community of animals that were able to live in this very low oxygen environment. And then we would go through that, and on the other side, we'd be going up into rising oxygen levels. And I just found this such a great experience every day to watch.
Definitely. I like to remind our audience to submit any questions you have, but I'm going to take moderator's prerogative and ask one. We all talked a little bit about the time and it takes to analyze samples and conduct your research once you're back in the lab. And I'm wondering what equipment on board would speed that up, if any. I know kind of eDNA and bringing that analysis to shore is a hot topic right now. And feel free to talk more about that. But if there's other technologies that helped you on board Falkor or would help you in the future speed up your processing once you're to shore, does anyone want to take that question?
Yeah, I mean, I can think of a couple of things. One of the things that we experimented with a little bit when in a collaboration through Schmidt was the use of the Squiddle software, which has a machine learning component to it that can start to analyze video basically as you're collecting it. And that is something that takes us a very long time that we go back, and Greg mentioned the AUV, or Lisa mentioned the AUV Sentry, and it takes tons and tons of photographs, and just that the processing time and the human component of going through all the video and photos and then trying to map out distributions, it's incredibly laborious, and having some of that automated would help. I think, you mentioned eDNA. I think we're at the point now where you can do DNA sequencing on board ships, and Greg talked about the importance of you can see these creatures and you can often tell the difference just from their morphology, their appearance, but that's not always possible. And a lot of the corals in particular, we have a really hard time telling apart, even if we're looking at two of them right there in our hands. And so having the ability to generate some of that sequence data on board and be able to analyze it to help identify these organisms immediately is going to go a long way. And I know both of those things are on their way, but that's going to be the kind of breakthroughs that I'll be looking for next. The tools we'll start applying.
Thanks, Eric. Does anyone want to add to that? All right. Eric's answer was comprehensive enough. So we did have an audience question about whether Falkor 2 is planning to return to Costa Rica, and I can answer that briefly. So we were planning to return with Falkor pre-pandemic, and unfortunately the world had other plans. But once Falkor 2 leaves the shipyard, which we're hoping will be later this year, we are hoping to be able to return to some of those sites that we had planned to during the pandemic and had to cancel. So I can't announce any cruise schedule right now because we're still in the shipyard and busy at work refitting the new vessel, but we do hope to make up some of those projects that we unfortunately had to cancel because of the world's events. But to turn that into a question for our scientists, if you were given the opportunity to return to Costa Rica and the national park and the boundaries that you worked in, what further exploration would you hope to do?
I can answer that.
Yes, please go ahead.
As you've seen, and everybody mentioned, it was an incredible expedition, but we saw only a little piece of what's there in this huge area. And as Greg put it very nicely, it was one seamount a day. And they were all different. It was, there were some things in common, but there was always something new. Every day there was something new. And we just saw a little bit. So there's still a lot to do, And I think it'll be interesting, which is not probably not very popular, is just to go out to the abyssal plains, to that muddy areas and do more sampling in there. Because from the few samples we took from these muddy areas, we found a lot of different creatures. And with a colleague from the University of Costa Rica, we published a paper on the fungi from here. And everything's new. And I'm sure with Victoria Orphan that's working on the bacteria, there's a lot of new stuff going there. And not only species, but processes that we just started to get to know. So it's a lot more to do.
Thanks, Jorge. Does anyone else want to add to that?
And I'll jump in.
Yeah.
The seamounts were really awesome, but we also went to some new seep sites on the Falkor cruise. And, you know, now that we can map seeps better with bubble plumes and, you know, there are all sorts of tools, probably, I don't know how many seeps have been visited off Costa Rica, maybe 10 or 15 total, maybe, but there must be hundreds. And I know of the couple of new seep sites we visited, you know, in 2019, they were full of different species and different assemblages of species than we had seen before. So I would think it would be really exciting to go explore more of the seeps on the margin. And also, of course, to run some oxygen minimum zone transects up and down and see how oxygen controls the community. We know oxygen minimum zones are expanding under global warming and climate change. And so understanding how they affect the ecosystem is going to be very important.
Sounds like an excellent research expedition between your and Jorge's plans. Eric and Greg, would you like to add anything to this expedition we're planning?
I just say people can look it up, but this mountain series of seamounts continues past Cocos and ends up at the Galapagos Islands. So there's this chain of ex-volcanoes that are ending up subducted under Central America. So there's the seamounts continue all the way out to Galapagos, and imagine the exploration of those. But as Jorge points out, there is just so many. It's an amazing opportunity for people in generations ahead, hopefully, to look at these. If we don't get the chance, other people will, I hope.
For sure. Greg, do you have anything, or Eric, do you have anything left to add? I'm sorry.
Yeah, I was gonna second that one. The other expedition that's planned is Beth Orcutt is the chief scientist and proposed that one. And it's to the Dorado Outcrop, which is a really interesting place that has warm fluids coming out, and it's a octopus nursery ground where you have just this huge high density of octopus all incubating eggs underneath them. And it's a really fascinating place, and I'm sure that expedition is going to be really interesting. And I just, I was really struck by, and Lisa talked about this a little bit, but how many creatures, big animals at high densities there were in the low oxygen zones. And this is, it is a major issue in the oceans because these oxygen minimum zones are expanding, and ocean deoxygenation is something that really Lisa awakened me to, and is something that's going to be a major issue that I think people aren't really talking about enough. And so these habitats and what allows some animals to thrive there is really a big open question that I think is going to be something that's going to be a big area of science in the future.
Yeah, thanks for those thoughts, Eric. Always something to think about, and you go out and you discover and you come back with more questions, I think is how science works, right? We have a question from Kat. I think this is for Lisa, but then I'll let others join in if they want to. Do you think Xenophiophore nurseries exist in other areas, or are they specific to this region that we visited?
Well, I honestly don't know, but I would suspect if we found two different fish species using Xenophiophores as nurseries, that this phenomenon probably occurs elsewhere, because snailfish are very abundant types of fish in the deep sea, and Xenophiophores are one of the only groups that are limited to the deep ocean. So, you know, there is some potentially some coevolution of behavior there. So I'm guessing that we should be on the lookout for this elsewhere in other
Great,
parts of the
thank
deep
you.
sea. And they're very fragile. It's probably worth noting that they're vulnerable to bottom trawling and other types of disturbance, being caught up in fishing line and things like that. And a few regions have designated them as vulnerable marine ecosystems, but you know, indicators, but most have not as VMEs. So I think we need to maybe rethink how we view Xenophiophores on the deep sea floor.
Yeah, and I didn't mention it earlier, but you actually had a scientific publication on that topic come out of this research expedition, but I think you have addressed all of those details. But if you would like to expand more on what was in the publication, please feel free.
Well, I'm trying to, Greg, you want to say anything about it? We basically identify, we're looking on the ship, opening up Xenophiophores, including some that had been in incubation experiments that Victoria Orphans Lab had been running, but and we found the embryos attached deep inside. So it was pretty clear that the fish had laid their eggs in there. And I think it's worth pointing out that snailfish have a habit of laying their eggs inside other organisms. So some will lay eggs in box crabs, some will lay eggs in sponges. I think they might even, I'm not sure about bivalves, but they use other animals to protect and nurture their embryos. So this behavior, it's the first time it was seen in Xenophiophores, but it's not that unusual as a general behavior for snailfish. I'm trying to remember like, was it Paraliparis? Greg, do you remember the taxa?
Yeah, but we weren't able to identify them any further because they were embryos, or you could see the picture, they were little tiny baby fish inside the egg membrane. But there are some DNA sequences of snailfish on GenBank, and that allowed us to prove that they were snailfish. But we couldn't go beyond that. We don't know if these were new species or not of snailfish, and so there it rests. But the DNA sequences are now lodged permanently on GenBank, and if anyone ever then finds the adults, they'll be able to then link up the story of the embryos in the Xenophiophores with potentially the adults. So that's one of the handy things about having a DNA archive available for people to access in the future.
Yeah, thanks, Lisa.
I'd like to add
Jorge.
something to this. Is that it's not only the diversity of the different creatures, but all those relations that we're looking at. This is a beautiful example that was mentioned, and the crabs and the black corals, the octopus and the wormer seeps coming up. It's not only looking at what's the name of this one, but all that connection, and what we're seeing is that they're all connected. And as Lisa said, the connection from the deep to the shallow waters, and it's amazing. And this type of expedition are great to start looking at that and be aware of that.
Thank you, Jorge. And thank you all for this great conversation. I love how we started off talking about Eric talking about what the goals of the expedition were and what each of you were working on. And I think we ended up with more questions that we discussed of your ongoing analysis than we started when Eric talked about what the goals of the program were and what he intended to do going out to sea. So it's a great example of how science works in general, just here on Zoom. I think we're almost out of time. And so I just wanted to give each of you a chance to say some wrap-up words about what you're working on now or what ongoing analysis is. I know we've touched on that in a lot of this discussion, but if there's any last forward-thinking thoughts you'd like to give to the audience before we sign off, that would be great. And I'll start with you, Eric.
Yeah, I mean, I started off saying this isn't an overnight endeavor, and it certainly continues. And I know that all of us will continue working together and hopefully have a chance to get back to Costa Rica. Yeah, there's so, like you said, there are so many questions, especially when you get a chance to do some really pure exploration like this. You never know what you're going to find, and you don't know what questions to ask. One of the discoveries we made that we haven't talked about yet are corals that were living very, very close to some of the seeps. And in fact, some of them that we brought up, they were relatively small, but they had these filamentous bacteria that were growing all over them. And a perfectly healthy coral, which I assumed it would be dying and decomposing, and it had all these huge furry bacteria that were almost longer than the coral itself. And we did some experiments on board the Falcor that showed that the coral were taking up this methane from the bacteria. And that's something that one of my graduate students, April Stavins, is still working on. And it's going to be a really interesting story. It's a phenomenon we've seen a few different places, but not quite as intricate as we found it here. So I'm looking forward to seeing how that comes out.
Yeah, we'll have to have a part 2 of this discussion to learn more. Jorge?
During this expedition, I had a chance to collect a lot of sediment cores, which are being processed now. So there's a group from geology and from biology from the University of Costa Rica looking at the foraminifera radiolarians in the sediments, but also the fungi, and we'll be looking at other groups there. Also recording the minerals there and the amounts of carbon that are being trapped in these deep areas, which is a big question of also what at least a mention of that carbon just going into these very deep areas and being removed from the atmosphere. So a lot to do.
And I heard you were coming out of retirement to work on all of that, right?
Well, I say this to work without interruptions of meetings and faculty meetings and things like that, and lectures.
Nice.
Right.
Lisa, would you like to go?
Sure. Boy, we're still working on all kinds of material. I'll give a shout out to Olivia Pereira, my PhD student, who's working on sets of experiments that we haven't even had a chance to talk about. We were able to do rock transplants and colonization experiments, all sorts of manipulative experiments. And the Sebastian helped collect a lot of those that we had put out the previous year. So with these experiments, we're really trying to understand the fingerprint of methane and how it is methane and the energy and biomass associated with it are translated to the surrounding deep sea communities. And so Ollie Ashford, previous postdoc, had been working on and defined the term chemotone, which is the transition zone between the methane seep and the background community. And Olivia is continuing to look at that and try to understand how the animals use methane and how they adapt to changes in access to the seep and so on. So there's still a lot of work ongoing on that.
Great to hear. Greg?
Well, I'm excited that we are wrapping up a biodiversity inventory of all of the species we discovered over the previous expeditions, culminating in 2019 for the seeps. So Charlotte Said, the collection manager here at Scripps, has a cast of many, many authors, including all of us on this call, documenting all of the animals we found on all of our cruises, linked with the DNA barcode sequence. Many of them are named species. Some of them are still yet to be named. And then for the seamount journey out to Cocos, I am looking forward to some really spectacular new species that are independent of that study that will be described in the next few years, and no doubt will be shared by Schmidt Ocean Institute. Looking forward to getting those out of the way one of these days.
Thanks, Greg. You provided a great opportunity for me to close by saying to all of our viewers, please follow us on social media, where we will continue to update you on not just these research outcomes, but other research outcomes from our scientists that have sailed on board. And we'll keep you updated on new cruise announcements and the progress of Falkor too. So with that, I would like to sincerely thank all of our scientists for joining us and wish them best of luck and best of analyses as they continue all of this work. And we look forward to hearing and learning more about their results as time goes on. So thank you everyone for joining us.
Thank you.
Thanks, Allison.
Thanks.
Thanks for hosting this. It's great.
Thanks
you for the
everyone. Thanks
invitation.
for joining.